So this is the overview of glycolysis. All right, you should be comfortable with what happens at each step, and you should be able to tell me, what happened between 3PG to 2PG, for example. Okay, so things like that. You should be familiar with the reactions that we talked about in glycolysis. Okay, so this is the overall overview of glycolysis with the EMP pathway. Right? Now, let's take a brief look at the thermodynamics of glycolysis. This is the 10 steps. I know the numbers are a little small for you to see, but what you'll notice is that with the exception of PGK, which is step 7, it just so happens that the energy for 1,3-BPG going through 3PG producing ATP is pretty much the same as the reverse, okay, with the exception of this step. All the other steps associated with ATP consumption or generation are the regulated steps in the pathway. Why? And that is because those steps are the steps where we supply extra energy to sort of make it favorable. Okay, for example, if you will look, the first reaction, glucose to glucose 6-phosphate, delta G standard, it's about -17. On the further right here, delta G, without any notation on it, basically this is accounting for people have measured intracellular concentrations of ATP of glucose, so it's closer to the real delta G that I always talk so much about. Okay, remember that it's the real delta G that determines the direction of your reaction inside the cell. Okay, it's the real delta G. So if you look at the real delta G for reaction 1, it's approximately -30. Okay, so it's very large. And then the third reaction, where you have the phosphofructokinase turning F6P into F16BP, the real delta G is -22, so it's fairly large. And if you were to look at all the other reactions, some are close to 0, some are just very small. Okay, and the other largest energy is your pyruvate kinase, which you go from PEP to pyruvate. That is because PEP is very high in terms of energy. So when it generates ATP, it still has so much energy such that the delta G real becomes -16. Okay, and you have 2 of those, so it's in parentheses -33.4. Okay, the bottom line is, remember I said at the very beginning of our class that if you were to regulate a pathway, you are going to control wherever you have large delta G changes, right? So these 3 positions are going to be your regulated steps, which we'll talk about maybe later, right? So these irreversible steps, okay, they're called irreversible because they have large decrease in delta G, okay? Typically, if you see maybe like -20 something or above -15, it's pretty much irreversible given normal cellular concentrations, but there are some exceptions. Okay. Now, recall that irreversible steps are places for metabolic control. So if you were to look at the energy change on the left side, what you notice is that here one large change, another large change, and then third large change. So these are the steps that we're going to regulate glycolysis. All right, before we talk about glycolysis, I really want to talk about the fate of pyruvate. Okay, what happens to pyruvate, right? Because, as we're talking about metabolism, we study how glucose gets consumed in glycolysis and the net product is pyruvate. But then maybe you're wondering then, like, what does pyruvate do? Okay, what does pyruvate do? Pyruvate, okay, it's a magical metabolite that can produce pretty much everything. Okay, so it's like it can go everywhere. First thing, pyruvate. Okay, so pyruvate is right here. Pyruvate can return upstream to central metabolism through this process called gluconeogenesis. We will talk about that. So pyruvate can go back and make glucose. Okay, and obviously glucose phosphates can then make cellulose, that kind of stuff. So pyruvate can go back. Okay? Pyruvate is also the precursor to many amino acids, and amino acids are precursors to proteins and nucleotides. Okay? So pyruvate can be used to make amino acids. Pyruvate can then also become oxaloacetate, which it goes down to here. Okay? So it can form oxaloacetate. We will talk about all of these reactions. When it becomes oxaloacetate, this is the TCA cycle intermediate, okay? And it can be used to produce some amino acids from here, okay? Or pyruvate, a large percentage of pyruvate will actually go to this thing called acetyl-CoA, okay? And acetyl-CoA can then be used primarily for three things. One, acetyl-CoA can also be used for amino acid biosynthesis. Two, probably one of the most important function for acetyl-CoA is that acetyl-CoA is the precursor to making fatty acids. Okay? Without fatty acids, you don't have cellular membranes. Right? So acetyl-CoA is used to make fatty acids. And then lastly, acetyl-CoA is used to fuel the TCA cycle, okay, or the citric acid cycle. And where acetyl-CoA gets oxidized into CO2, in the process, it generates a lot of NADH. NADH is then used to produce ATP in this oxidative phosphorylation chain. Okay, this is electron transferring chain. Right? So pyruvate is sort of the center for almost everything. Okay? We will talk about later why acetyl-CoA cannot go back to pyruvate. Okay? So acetyl-CoA cannot go back to pyruvate, at least in humans. Okay? In bacteria, yes, it can, but in humans, acetyl-CoA does not go back to pyruvate. Okay? Actually, this is the reason why you gain a lot of weight if you drink alcohol too much. Okay? Because alcohol is 2-carbon alcohol, right? Ethanol. And you drink a lot of alcohol, ethanol gets metabolized. Ethanol coming in, it becomes oxidized, become acetaldehyde. Acetaldehyde gets oxidized to become acetyl-CoA. Okay? But acetyl-CoA cannot go to pyruvate. So acetyl-CoA can only go to two places, where one is TCA cycle, one is fatty acid. Of course, you have amino acids, but your body does not make a lot of amino acids. It makes a lot of fat. Okay, so yeah. So if you drink a lot, you're going to gain weight. Okay, because that's just how it is. Right? You'll gain storage. Your cell wants to store these acetyl-CoAs. Okay, so fate of pyruvate is a lot of places. And now that we cover glycolysis, later on you will see that pyruvate can do a lot of different things. Now what we're going to talk about today is that pyruvate can be used in fermentation, which it can be used to produce lactate or ethanol, okay, which have significant importance to both the industry, or it has a lot of socio-economical values. Okay. Now, the first thing is actually I want to talk about is fermentation. Okay, fermentation is a very important process. What fermentation is that essentially it's a process that we use when we don't have oxygen. Okay. Now, when oxygen is present, okay, respiration can take place. So if you have oxygen, you can do respiration, and the NADH that you generated in the process of cellular metabolism will then pass the electron from NADH to O2, okay, to oxygen. In the process, you generate ATP. Okay, this is what we'll talk about. The key point being that NAD is regenerated, okay, because NADH, where is it? Glycolysis, glucose becoming pyruvate, generate ATP, and you get a bunch of NADH. And then NADH can then use oxygen as the electron acceptor to donate the electrons from NADH to oxygen, and you form water and NAD. Okay? So the most important process for respiration is actually you can regenerate NAD+. Okay? So now you regenerate NAD+, your glycolysis can continue. Makes sense? Okay? Now, if somehow you don't have O2, do I have animation? Okay. If you don't have oxygen, such as anaerobic microorganisms, so like for example in sewer treatment, like water treatment, like you go to the bathroom and you release a bunch of shit, and or your sink. Okay, you have wastewater. Okay, all these wastewater gets collected somewhere. Okay, and usually it gets collected in this pile of tank. Okay, inside it there are these bacteria or archaea. Okay, it has these microorganisms, and these microorganisms are then capable of digesting whatever that is in the environment. Okay, so it's kind of like a water treatment process. Okay. However, most of these organisms, turns out, they're anaerobic organisms. Why? Okay? If you think about like a tank underground, it's not going to have a lot of oxygen, right? So obviously, they're capable of doing some kind of metabolic process such that they can recycle NADH into NAD. Okay? So these anaerobic microorganisms are, capable of somehow regenerate your NAD and your glycolysis continue. Or intensely exercised muscle. So if you go and run like crazy, your body, actually your muscle parts are short in oxygen. Okay? That is because you're burning your oxygen very fast and your bloodstream does not carry the oxygen fast enough. Okay? So in those cases, how does your muscle respond to this demand of NAD in order for glycolysis to continue? Okay? That's sort of what we want to talk about. Okay, so NADH really somehow needs to find a place to dump its electron in order to generate NAD+. Glycolysis can continue. Okay, now turns out pyruvate can serve as an electron sink when O2 is not available. Okay, so when you don't have oxygen here, you don't have NAD, you have a buildup of NADH. Okay, so you have a lot of NADH, and your NADH has to do something, right? And turns out in humans, we produce lactate. In many bacteria, like yeast, they produce ethanol. Okay? NAD is then somehow replenished because you are now able to consume your NADH. So NADH returns to normal, NAD returns to normal, so then everything can continue that way. Okay? So in the absence of O2, a lot of organisms will initiate fermentation. Okay? This is fermentation. Okay? It's in the absence of molecular oxygen. Your cells will use internal metabolites and use them as the electron sink. Okay? So pyruvate becoming lactate or ethanol. So pyruvate will then react with NADH and form something else. Obviously, this is important for industry because pyruvate, if you want to make yogurt or like yogurt drinks or your fermented pickles, okay, fermented stuff, or when you exercise so much, okay, you're gonna produce lactate. So pyruvate becoming lactate is a very simple process where we only use one enzyme and it can directly reduce pyruvate and become lactate. Okay, so this is lactate pathway, and a lot of times there are these bacteria that can do this homolactic fermentation. So essentially this is like lactobacteria. They can take glucose and then turn it into lactic acid, okay, and then you can eat that. Or some bacteria are able, or yeast or bacteria are able to use pyruvate. You do a decarboxylation to form acetaldehyde, okay? And so here there's no oxidation or reduction, but then what you can do is take acetaldehyde and then reduce it to ethanol. So in the process, regardless of if you make lactate or ethanol, what you do is you're consuming NADH, okay? So that your cell can grow. And these reduced products typically have high values. Okay, lactate, you can eat it, or you can make polylactic acid plastic. Okay, so you guys know about PLA cups. If you go to Starbucks, and you order like a cold drink, and if you look at the bottom of the plastic cup, it says PLA. It's polylactic acid plastic. So it's a biorenewable and biodegradable plastic. Okay, so that's made out of lactate. Okay, so that's made out of lactate. Ethanol, obviously, you can use it to make alcohols, or you can make it into ethanol biofuel, things like that. So first, we take a look at lactate dehydrogenase. Okay, lactate dehydrogenase. You've seen this in your first day of class, where you turn pyruvate initially. This is a keto group. and you turn that into a hydroxyl group. Okay, so you are doing an reduction. Okay, so you reduced pyruvate using NADH and you form lactate. Okay, and this is quite favorable. And the reaction mechanism is kind of crazy, but it's actually a lot simpler than you expect. Okay, so you have seen how we generate, how we make NADH, right? In our reaction, I don't know, like 6 or something. You have seen how we make NADH. Now, at the end, your NADH has lone pair on the nitrogen, and what the nitrogen lone pair can do is it pop back down, this electron pop over here, and now this carbon has too many bonds, such that one of the hydrogen will leave as a hydride, so H minus. Hydride now is a very good nucleophile. It will attack the electrophile, which is the carbonyl center, okay? And here you'll form a, you pop the electron over to the oxygen and you form this intermediate kind of thing. Okay, you form this oxyanion, and oxyanion can then grab a proton, okay, grab a proton, and it becomes lactate. And turns out, when you go exercise a lot, and your body will generate a bunch of lactate, okay? So lactate produced in glycolysis during muscle exertion. Exertion is like, exertion. The lactate, after that, what do you do with it? Okay, so like it doesn't just sit there. So the lactate is then transported into the liver. Okay, and so this is muscle. Okay, in the event of exercising, your glucose is going to become pyruvate, and pyruvate will become lactate. Okay, that is because your muscle cells do not have enough oxygen. So then the muscle cells will say, hey, let's somehow recycle my NADH back into NAD, and to do that, I produce lactate. Okay, and then your lactate can then be transported by the blood and transported into the liver, where in the liver you will do gluconeogenesis. Gluconeogenesis, lactate becoming pyruvate, pyruvate becoming glucose, glucose can recycle. Okay, so lactate is not just secreted. Okay, you don't secrete lactate, just so you know. Lactate is recycled. Ethanol fermentation. Okay, ethanol is a very important product for humans because obviously we know how to ferment alcohol back when, like a couple thousand years. The way it works is that it's mediated by the ethanol production pathway. 2 steps. The first step is this thing called pyruvate decarboxylase, where you take this carboxylate, and you just kick it out as CO2, okay? And what you're left with is acetaldehyde. And this acetaldehyde can then be reduced, aldehyde be reduced to alcohol, and then you generate ethanol, okay? So these 2 enzymes. Currently, most common alcohol-producing microbes, primarily there are 2. One is Saccharomyces cerevisiae. This is baker yeast. You use this to make like beer, you use this to make bread, that kind of stuff. Or Ximomonas mobilis. This is a type of bacteria people use to make, I think wine, you use this, or some kind of other fermentation product. Okay? Also, this is a side note for you to remember, humans also have alcohol dehydrogenase, but it's primarily for metabolizing ethanol, like what I just said. Taking ethanol, you go reverse to produce acetaldehyde. Okay? And then ethanol is oxidized to acetaldehyde, which is then converted to acetyl-CoA and enter the TCA cycle. Okay? But if you take too much of that, you make fat. Okay? You make lipid. Pyruvate decarboxylase. Pyruvate decarboxylase is a very interesting reaction. I do have the mechanism. Pyruvate decarboxylase, what it does is it's a decarboxylation of an alpha keto acid. Okay? This is something that I want you guys to definitely remember. The difference between alpha keto acid and beta keto acid. This is an acid. Okay? That's acid. Okay? This is alpha carbon, beta carbon, gamma carbon. Okay? Alpha keto acid. So this thing is called alpha keto acid. Because alpha keto acid. Okay? And this other thing, if you shift the keto group to alpha, beta, gamma. This is beta keto acid. Okay. One thing about beta keto acid that you should know, okay, beta keto acid is generally not stable. Okay, it's generally not stable. Okay, why? Okay, the reason being, okay, it doesn't need to have an enzyme to catalyze this because here is your minus charge. If your minus charge comes back here, okay, and essentially you can form like a new bond here, and then this is essentially you can sort of and this will kick out a CO2, okay? You can form this enolate, right? Enolate can come down, this can grab any proton, okay? So the reason why this is not very stable is because you have resonance on here. Okay, you have a resonance structure where this thing can easily leave. Okay, and the immediate product has resonance structure where you sort of can form. You sort of can form this. Okay, kind of like that. I don't want to draw it like that. I just leave it as this. Okay, you have a stabilized enolate, and that can easily grab a proton in the solution. You can easily grab a proton in the solution, and then what then you generate is CO2. Okay, so beta keto acids in general not very stable, not very stable. Okay, so if you take this structure and you look at it, it will spontaneously decarbosylate to acetone. Here, alpha keto acid is different. Okay, it doesn't like if you do this doesn't have anywhere to go. Okay, it doesn't like go anywhere. Right, so alpha keto acids in general is more stable. It's slightly more stable. Okay, so this is a very important thing that you have to know. Beta keto acids favors the decarbosylation. Alpha keto acid does not. Okay, this is the key. Pyruvate is an alpha keto acid. Okay, so if we want to do decarbosylation, it's not going to be very easy unless we somehow make it into a beta keto acid. Okay, so if you look at pyruvate, this is alpha keto acid. So alpha keto acid, if you want to do decarbosylation, it becomes like that. And you form this really, really unstable carbanion. So that doesn't happen. Okay? This is the reason why alpha keto acid does not spontaneously decarbosylate into its decarboxylated product. Okay, so turns out this reaction, if you want to do an alpha keto acid decarbosylation, you need to use the cofactor called thiamine. Okay, now thiamine is vitamin B1, right? And this thiamine becomes thiamine pyrophosphate. Oh, sorry, this is the active cofactor. This is the precursor that we eat in our food. Okay, so thiamine pyrophosphate is the coenzyme or the cofactor used by most of these decarboxylases that can do alpha keto acid decarbosylation. Okay, so vitamin is structurally complex, so this we can take from our daily diet. But in order for this to become active, it has to be first activated using ATP to become thiamine pyrophosphate. Okay, and the part that functions for the decarbosylation is highlighted in blue, is this part. The thiazole ring of the TPP, this is called TPP because thiamine pyrophosphate, TPP, okay? TPP, the thiazole ring for TPP is the functional part for this coenzyme, right? So that's how it is. All right, first, let's look at how TPP gets activated. TPP is like this, right? So if you look, here is a carbon, it's a 5-member ring. 3 of the member are carbon, and 1 is nitrogen, and 1 is sulfur. Okay, so like that, like that. The first thing is, what you can do is do a deprotonation such that you can generate this TPP anion, kind of, this E-lit form. And this anion can then attack the carbonyl center on the pyruvate, okay? Such that what you generate is this oxyanion type of intermediate. And this oxyanion grabs a proton from the solution. Okay, so this guy attack here. And then what you get is O minus, right? O minus, grab a proton, it becomes this. And then if you were to look at this structure, okay, this in a sense is an beta keto acid. Okay, why? Because you have the carbonyl, this is the alpha carbon, this is the beta carbon. Okay, the beta carbon now has a electron withdrawing group. Okay, just like your beta keto. Okay, so because of that, what you can do is the electron pops back down, this guy pop over here, okay, and then you eliminate the CO2, right? Although you form an alkene over here, what you can then generate is that you form this double bond, this E-lit thingy, and then you can then put 1 electron back onto the carbon, and this carbon becomes carbanion, okay, like that. And this then can then grab a proton like this, and then your OH claps back down, kicking out the TPP, you generate acetaldehyde. Okay, so this is the catalytic cycle of TPP-mediated decarboxylation. But what you should notice, that the key idea is that over here, you generated a beta keto acid, kind of, okay? Remember that here, this is the electron withdrawing mechanism so that you can stabilize the electron from decarboxylation. Okay? The electron can be stabilized here through resonance. Okay? So that's the key idea. So turning alpha keto acid into a beta keto acid such that you can mediate the decarboxylation. Back in, if you don't have TPP, okay, you don't have anything here that can withdraw the electron. Because of that, this intermediate is very unstable. Okay? If you have an unstable product, that means you have a stable reactant. Okay? Here you have a more or less stable product, therefore you can then sort of think of the reactant as more or less unstable. Okay, so everything stable, unstable, it's a relative term. Okay, it's a relative term. So that's the way to think of it. Okay, and then you can see how once you kick out the CO2, and then you just have to somehow pump the electrons back in, and then at the end of the day, you get acetaldehyde. Okay, so when we talk about fermentation, obviously you should be thinking in terms of aerobic versus anaerobic. So aerobic is aerobic exercise. That means that you're in the process of having a lot of oxygen. Okay, so aerobic is something with oxygen. Anaerobic is an environment where you don't have oxygen. Okay, now it says here that generating ATP. Okay, I'm missing a word here. ATP from glycolysis is much faster than from respiration. Okay, however, with much lower ATP yield. Okay, so at the end of this first third of our class, you should be able to understand what I mean by this statement. Okay, now muscle cells utilize this fast ATP generation when undergoing intensive exercise. Okay, intense or intensive exercise, and basically, that's what we just talked about, how a muscle cell, because it doesn't have O2, so it really needs to have a fast way of generating ATP. Therefore, it chooses to produce lactic acid. Now, what's interesting is that cancer cell can achieve higher or high or higher growth rate through metabolizing glucose and producing lactate even when O2 is present. Okay, so normally in your muscle cell, like you're just sitting here and your muscles are properly filled with O2, okay? You're generating ATP at a very good rate for whatever you're doing, which is sitting here. Your muscle don't need to produce lactate, okay? You're not just randomly producing lactate for the heck of it, okay? You don't do that unless you're exercising. But turns out in cancer cells, you guys know that cancer cells are sort of these cells that are growing without control, okay? And they have random mutations, and some cancer cells turns out that they can generate lactate even when O2 is present. So what that means is that you will be generating a lot of ATP just uncontrollably, and as a result, you promote faster growth. Okay, that kind of makes sense? All right, so in the aerobic process, okay, glucose, regardless of aerobic or anaerobic, glucose first get down to pyruvate. And in the aerobic process, we will take pyruvate and we're going to do this process called the citric acid cycle, the TCA cycle, which we'll talk about in our next class or the next class. And what then this TCA cycle do is it's going to turn pyruvate, which is sort of organic carbon, okay, organic carbon, into CO2. Okay, so it's going to turn pretty much all of the carbon here into CO2. And then in the process, you take out the electrons stored in pyruvate and you put them onto NADH, okay, and FADH too, but primarily NADH. And then NADH is going to use this process called the oxidative phosphorylation, okay, and then this thing generates a lot of ATP, okay, and you know this process just continues and on, okay. So that's the aerobic process, and in the anaerobic process, you don't use this oxidative phosphorylation. Instead, you're going to use homolactic fermentation producing lactate, or for bacteria, you produce alcohol, okay? All right, so aerobic, it's slower, but very good energy yield in terms of producing ATP. Anaerobic, typically fast, but lower energy yield. Why is that? What do you guys think? Why is it that anaerobic process, this process, compared to the aerobic process, why is this process lower in energy yield? By energy here, I mean ATP. Energy yield meaning how many ATP I can produce, right? Okay, and ATP is going to probably couple to, I don't know, oxidation of organic products, right? So it turns out, the reason why your anaerobic process, it is definitely lower in energy yield. That is because you start with organic carbon, you still end up with organic carbon, okay? Whereas in the aerobic process, you have organic carbon, okay? And then you turn it completely to CO2. So all of the energy in pyruvate is stripped out, okay? It's taken out from pyruvate so that the carbon in pyruvate becomes CO2. And remember, CO2 does not have energy for biology, okay? Okay, but on the other hand, anaerobic fermentation, you're left with another organic product. And because of that, you still have energy stored in, for example, lactate or ethanol. Okay? Does that make sense? Right? So like I said, if you take your muscle, okay, some people are just naturally fast runner. When you were in elementary school, your teacher probably told you that if you run slow, it's not really your fault, it's genetic. Okay, so if you're a slow runner, okay, if you run like super slow, it's not your fault. Okay, even if you train, you're not going to be very fast. Okay, that's just how it is. Okay, yeah, certain things are born with it. You know, this is because if you were to take a slice on your muscle cell and you look at the distribution of the muscle cell, and here you'll see that. Let me see real quick. You'll see that here on the left side, this is slow twitch muscle fiber. Okay. It has a lot of red. Okay, obviously the picture is black and white, but I'm going to tell you this is red, this is like white. Okay, where on the other side, on the right side, you have fast twitch muscle fiber. Okay, which you have less red cells and more white cells. Okay, turns out, more red corresponds to more heme. Okay, more heme, and heme is sort of related to like hemoglobin, related to transfer of O2, that kind of stuff. So basically what this means is that these red cells are going to have a lot of heme-containing mitochondria. Okay, so they're going to use mitochondria, they're going to use respiration for energy generation. Okay, so they are going to use O2. Okay, these cells are going to use oxygen. On the other side, where you have fast twitch muscle, they have less cells with a lot of mitochondria, so you essentially have fewer mitochondria. So you have a heavier reliance on the anaerobic metabolism. And remember, the anaerobic metabolism is faster, okay, it's quicker. The process is quicker. So that turns out your fast twitch muscle fiber can generate ATP faster, okay? And then you use ATP to have muscle contraction so that you run faster, I think, okay? All right, so that's sort of a good example of how this thing works. All right, so that's sort of pretty much the extent of fermentation that we're going to talk about. That's pretty much it. Do you have any questions on fermentation? Fermentation is actually a very important aspect in biotechnology. Okay, so if you guys are interested, you can come talk to me individually, or I have a class next semester that is focused on how to engineer metabolism to produce a wide variety of different products. Okay, so fermentation is being used by modern day scientists and engineers to produce all kinds of chemicals. Okay, you can think of chemicals ranging from plastics to amino acids to fuels to whatever. Okay, and a lot of those processes are related to fermentation. Okay, it's a very powerful mechanism. All right. So the next general topic we're going to talk about is this thing called gluconeogenesis. Okay, gluco means sugar. Genesis means like to generate, to synthesize. Okay, so this is glucose biosynthesis, sort of. Okay, so you can synthesize glucose. Well, first of all, why is gluconeogenesis important? Okay, I mean, why? Right? That is because if you think about you as a human and you have a daily diet, let's say you go on a diet of you eat only proteins, okay? You eat only proteins. You don't eat any sugars, okay? Any carbohydrates. But then, I don't know if I told you this last semester, but in humans, our short-term storage of energy is glycogen, okay? Glycogen is polymer of glucose. How do you convert amino acids to glucose? Okay, amino acid gets degraded to pyruvate. Pyruvate then convert itself to glucose. Glucose then make it itself into glycogen. Okay, so that's how you store it. Turns out also many organisms and many cell types require a constant supply of glucose. For example, neurons. Okay, your brain basically eats only two types of food. The first type is glucose. Okay. The second type is this thing called ketone body. We'll talk about ketone body when we get there. But your brain does not like to take ketone bodies, right? Which is why if you are sort of short on glucose, you don't get very happy, right? You have a cranky mood, right? You're going to be very upset. Which is why a lot of people, if they drink something sweet, you can sort of have a satisfying feeling, okay? Believe it when I say when you study in your PhD program, when your advisor is really annoying, right? You're gonna start to drink a lot of sugary stuff. Okay, that's how I gained like 10 kilograms. All right. Anyways, yeah. So your brain really likes glucose. Okay, so some cells are just specific to certain food. Okay, and your cell, your nerve cells, or at least your nervous system, they like glucose. Okay, they don't eat amino acids. Okay? They don't eat fats, right? They want glucose, right? So here, what I say? Brain, skeletal muscle, kidney, blah blah, erythrocytes. They use glucose as their sole or primary energy resource. Okay. However, turns out in your brain they lack the enzymatic machinery to synthesize it. Okay, that's interesting. And sort of little information for you guys to know: the kidney, okay, your kidney, part of the kidney. And your liver, obviously. Liver and kidney are the primary gluconeogenic tissues. Okay, so you synthesize glucose in your liver. Okay, like what we saw in CoreCycle. All right. So in humans, glucose can be synthesized from pyruvate or lactate or oxalacetate or some amino acids. Okay, not lipids. Okay, this we'll talk about later, but not lipids. Okay. Through this pathway called gluconeogenesis, mainly occurring in the liver. This is particularly important for converting human diet intake to glucose, somehow turning a big ass steak into Coke with a lot of sugar. Okay, magic. Okay. Bottom line is that gluconeogenesis really provides the flexibility in metabolism. Okay, it's the ability to interconvert different types of molecule. That is really important because when we're talking about biology, life, okay, one of the very essential sort of feature for metabolism is that you have to be flexible, right? You have to be able to interconvert different things, okay? And lastly, probably, for me, really, I think the most important part of gluconeogenesis is that, of course, this is a hypothesis, but scientists really, some of the scientists think that nature evolved gluconeogenesis before glycolysis, okay? And it sort of makes sense because back when, eons ago, okay, Earth was not organic, okay? Earth has a lot of carbon monoxide, methane, hydrogen, CO2, that kind of stuff, right? And we talked about emergence of life, where we talked about how like RNA was kind of the first molecule, okay? So it's not going to be carbohydrates, okay? Now, the initial organisms probably evolved to utilize methane, CO2, and carbon monoxide. Now, these things are not glucose, right? So then through evolution, gradually organisms evolved to then convert these things into carbohydrates, okay? And obviously one of the reasons you can think of is that carbohydrates can form useful things for structure, okay? Like cell walls or your, what else? Your like crab shells, that kind of stuff, right? We talked about carbohydrates being a very good material for making structures, right? We did, okay? So that's probably sort of the evolutionary route for converting simple molecules like carbon monoxide and CO2 into something like carbohydrates, okay? And then gradually, when you have a lot of carbohydrates on Earth, okay, if you have something, all right, some other organism somewhere is going to evolve to use it, okay? So the world as we see today obviously is balanced. I mean, today maybe not so balanced because of humans, okay? Like we create a lot of trash. But in general, evolution is sort of reaching to a state of balance. There's nothing that's going to net accumulate, okay? If you net accumulate glucose, you're going to be filled with sugar all everywhere, but that doesn't happen, right? All right, so gluconeogenesis. If we look at the stoichiometry here, you see that we can consider the exact reverse of glycolysis, where we take 2 pyruvate plus 2 ATP plus 2 NADH to generate glucose, ADP, and NAD, okay? The reverse reaction of glycolysis. Then you're looking at a standard delta G of plus 83.1, okay? Yeah, that doesn't happen, okay? This doesn't happen, right? Because this pathway would be very thermodynamically unfavorable. Okay, so it doesn't really happen. Right? We know that glucose go to pyruvate minus 83, very good. But the reverse, the exact reverse, is not very good. Okay, so it turns out, gluconeogenesis is going to invest in additional ATP. Okay, so here turns out we're going to consume total of 6 ATP equivalent. It says here 4 ATP and 2 GTP. Again, remember ATP, GTP, CTP, and what am I missing? TTP. They're triphosphates, they're nucleotide triphosphates. They have similar energy, so we can sort of lump them together and say they're pretty much the same thing. Okay? So gluconeogenesis occurs through 2 pyruvate plus 6 ATP plus 2 NADH. Okay? And that will give you glucose, and then you would change sort of the delta G into minus 33 kilojoule per mole. Now this process will become favorable. Okay? So you have to invest in additional energy in order to make the pathway efficient. Right? Remember, most metabolic pathways have to have a net negative delta G. Okay? Otherwise, it's not going to happen. Okay? So if you look at the two equations, the top one and the bottom one, what you see is that you consume the same amount of NADH. Okay? Here I'm going to tell you, you have to consume the same amount of NADH. Can somebody tell me why? You can consume different amount of ATP, but you have to consume the same amount of NADH. Take a guess. 为什么你的就是同樣是2個 pyruvate 到 glucose？上面是吃2個 NADH， 然後下面也是一定要吃2個 NADH。我說一定哦 ，OK， 一定哦。如果你今天要把 pyruvate 變成 glucose， 但你一定要用2個 NADH， OK， 就是2個 pyruvate 到1個 glucose， 你一定要用2個 NADH， 或者是 NADH 的 equivalent。為什麼？ It has exactly to do with oxidation reduction. OK， 因為 electrons. Okay, remember that electrons cannot be created nor destroyed. You learned that in like physics. Okay, you cannot just magically create electrons, right? Nor can you just randomly destroy electrons. Okay, electrons have to be sort of conserved. Now, if you go from pyruvate and you want to go to glucose, okay, you have to use 4 electrons from 2 NADH. Okay, you have to, right? ATP, remember, is used to make your reaction more favorable, okay, because it has to do with enzymes mechanism. But ATP doesn't have to be conserved. NADH has to be, okay? So from same reactant to the same product, the number of NADH, okay, the number of electrons, I should say, will always be the same, okay? Provided that you don't have any byproducts, okay, then it will always be the same. So gluconeogenesis, if you were to compare glycolysis, so on the left side here, it shows glycolysis. And glycolysis take glucose, and then you consume 2 ATP here, and then you produce some ATP, right, and then you generate pyruvate. Now, when you go back, okay, so going from PEP to pyruvate, okay, while it produces ATP, and the energy is still minus 30. Okay, so here is if you consume ATP, the energy will be plus 30. That doesn't work, okay? So we're going to have a different mechanism for converting pyruvate to PEP, right? I mentioned there was a exception here with 3-PG to 1,3-bisphosphoglycerate, using 3-PG plus ATP to 1,3-BPG. This one is reversible, okay, because it just so happens that the energy were okay. And the other two, okay, here you have fructose 1,6-bisphosphate going back to fructose 6-phosphate, okay. If you were to produce ATP, okay, then you have a delta G of positive like 16 or something. So instead, we're going to use just water to hydrolyze out one of the phosphates, okay. So remove the 1-phosphate such that you can get F16BP to F6P, okay. And then same thing for glucose 6-phosphate to glucose. Okay, so you just use water to do hydrolysis, removing the phosphate, and guess what? Phosphate is very stable, so the product is stable. Reactant is less stable, so delta G is minus. Okay. All right. So here I highlight the steps that are different in glycolysis from gluconeogenesis. Okay. So 3 steps. Basically, all of the ATP consuming or producing step except the 1,3-BPG step. Okay. So all of the ATP steps are going to have a different enzyme. Okay. Now, here, going from pyruvate to PEP, there are different types of enzyme. In humans, we're going to use 2 enzymes. We're going to use pyruvate carboxylase, and then we're going to use PEP carboxy. Hold on, this doesn't. Right. Okay. So pyruvate carboxylase. Okay. We'll show you in the next slide. Take pyruvate to oxaloacetate. We'll show you the structure in a minute. Okay. And then going from oxaloacetate, you can then make phosphoenolpyruvate, okay, PEP. So it's in 2 steps. Okay. Now in E. coli or some other bacteria, there's this thing called pyruvate synthase. Okay. They can use pyruvate to PEP by using ATP to AMP energy. Okay. And remember that ATP to AMP has more energy than ATP to ADP. 就是 ATP 如果 generate AMP 啊 ，OK， 它其實能量会比 ATP 到 ADP 来大嘛 ，OK， 有齁 ，OK， 那其實一個 ATP 變成一個 AMP 啊，其實等於啦，你等於吃了2個 ATP。有沒有要問我 why？ 沒有人要問我 why， 對不對？不 care 齁。為什麼？對不對 ？OK， 這是一個非常好的問題 ，why？ OK， why？ So ATP, typically we say ATP, you consume ATP, right? You consume 1 ATP. We generally mean it by saying ATP becoming ADP and then, PI, right? Now, if you go from ATP to AMP, okay, monophosphate, okay, monophosphate plus, I don't know, PPI or something, okay, and then PPI can then become PI plus PI, something like that, okay? Now, the reason why I say it's essentially the same thing as consuming 2 ATP is because AMP, in order to generate AMP back to ATP, okay, so here essentially you can go back by, let's say consuming 1 ATP, okay, so you can transfer the ATP from ATP to ADP, but it doesn't really make sense. We'll talk about how you generate ATP a couple lectures later. But here, in order to get AMP back to ATP, you need to first take AMP back to ADP, okay, to the diphosphate. Here you use ATP, okay, and then go from ADP to ATP, and then let's say you use another ATP, something like that, okay. Yeah, I'm just not going to write this ATP thing, okay. Right. So essentially, if you take 1 ATP to 1 AMP, okay, this is the same thing as saying here's 1 ATP, here's a 2 ATP. So this guy is the same thing as saying 2 ATP, okay, to 2 ADP, okay, because ATP come in, you produce ADP. Make sense? Okay, so basically if you see a reaction that converts ATP into AMP, that's the same thing as consuming 2 ATP. Gluconeogenesis, reaction 1, pyruvate carboxylase. Okay, it's a carboxylase. So it's going to do like a carboxylation reaction. Okay, so it's going to grab a CO2, or in this case bicarbonate, and then it's going to put it on your molecule. Okay, this is one of the few enzymes that you use to take in CO2. Right? Pyruvate carboxylase. Okay, pyruvate carboxylase. You take pyruvate, which is the alpha keto acid, like this, and you would use ATP energy, and you grab a bicarbonate in the solution. And remember, bicarbonate is nothing more than just CO2 plus water. You get carbonic acid. Carbonic acid can become bicarbonate plus proton. Okay, so it's this guy. So pretty much the same thing as CO2. All right, so here you grab a bicarbonate or CO2, whatever. You grab a bicarbonate, and it's going to hook it up, guess what, to your alpha carbon right here. Okay, this is acid alpha keto, and that's the beta. Okay, and then you put it onto the beta carbon. Okay, and pyruvate carboxylase contain a particular molecule called biotin. Okay, biotin is vitamin B7. So we talked about vitamin B1, we talked about vitamin B7. So you can go home again to show your mom, hey, yo, vitamin B7 is this, okay? And it's used for, I don't know, doing gluconeogenesis, okay? You could be like, eat more biotin. Biotin sort of serve as a CO2 carrier, okay? Just like NAD is a hydride carrier, biotin is typically the CO2 carrier, okay? Let's look at the mechanism real quick, okay? Biotin looks like this. And the active part of biotin is actually on the carbonyl and then two nitrogen attached to either side. And here you have a lone pair right here. What is going to happen first is that this is ATP, right? He'll show you here is phosphate, and that's ADP, right? So then you're going to sort of convert your bicarbonate into an activated bicarbonate by first taking bicarbonate and you phosphorylate that, like phosphorylate bicarbonate. You phosphorylate bicarbonate, so you have a bicarbonate attached to phosphate, okay? And then you can have biotin, the lone pair, attack the carbonyl center of your activated bicarbonate, and then essentially what that happens is you'll grab the CO2 moiety right here, CO2, okay, onto bicarbonate. You form this intermediate, okay, this bicarbonate on biotin, And then what that then happens is you have a mechanism which you can deprotonate the hydrogen here on the beta carbon. And obviously this is deprotonatable because here is a carbonyl, so that's alpha carbon in relation to this guy, okay? So then you can pull off that particular H+, okay, deprotonate this carbon, and then you have a carbanion here, which that can then attack this electrophile carbonyl center, electron jump over back to biotin, and then you have your oxaloacetate. 你看那個 arrow 就知道了，好不好？就是你会先把 bicarbonate， 然後利用 ATP 的能量把这個 bicarbonate 做 phosphorylation，OK， 做 phosphorylation 以後，你的 bicarbonate 能量就比較大。這個時候你的 biotin 可以去 attack 你的這個 activated 的 bicarbonate。之後呢，你會把那個 bicarbonate 等於有點那個 bicarbonate 上面的 phosphate 踢掉 ，phosphate 很 stable， 所以這個 reaction 變成 favorable，OK，and then 你會得到這個 biotin 上面插了一個 CO2， 然後這個時候呢，你的 enzyme 會去把 pyruvate 後面這個 methyl group 上面的其中一個 hydrogen 拔掉，變成一個 carbanion， 它就會變成一個 nucleophile attack 一個 electrophile，OK， 然後就變成 oxaloacetate，OK，makes sense。All right. Now, oxaloacetate can then become phosphoenolpyruvate, PEP, and essentially in this step, both the decarboxylation and GTP hydrolysis are used to provide energy for making PEP. Remember that PEP has a huge amount of energy, right? So you need to have a very good thermodynamic driving force. Okay, one of the driving force is we're going to consume ATP. Okay, we're going to consume ATP. Another driving force is decarboxylation. Okay, why is decarboxylation a very good thermodynamic driving force? Because CO2 is a gas, it leaves the system. Okay, because this guy is a gas, so it leaves the system. So then you're always pushing into this direction. So CO2 is usually any step that kicks out CO2, it's usually very favorable. Okay, and typically irreversible. Okay, and the mechanism looks like that. Okay, so you have some, this guy sort of pop back down, okay, and then you form the alkene, and then here, the electron is going to grab a phosphate. Simple as that. Oxaloacetate is a alpha keto acid or beta keto acid. I say alpha. Okay, why? So it turns out this molecule is both beta keto acid and alpha keto acid, right? So in relation to this guy, reference to this guy, okay, you have alpha beta keto acid, okay? But in reference to this guy, this is alpha beta, both alpha keto acid and a beta keto acid, okay? So if I want you to draw the beta keto acid decarboxylation product, what would you draw? Okay, that's something for you to think about. Okay? All right. So gluconeogenesis, really reactions 3 and 4 are phosphatases. They're just nothing more than just water come in and hydrolyze your phosphate away. So these two. Okay, so fructose 1,6-bisphosphate to F6P, G6P to glucose. Okay, so these phosphatases, they're called phosphatase because it's a phosphate, ACE, phosphatase. Okay, so they degrade phosphates. Right? They get rid of the phosphate. So then you can start from pyruvate and you end up with glucose. Okay, so this is gluconeogenesis. All right, regulation. All right, so I can't just be talking about the chemistry. Okay, I need to talk about regulation, even though I don't really like regulation. Now, gluconeogenesis and glycolysis, they don't happen at the same time. Why? What happens if they do? Glycolysis 呢，为什么不能跟 gluconeogenesis 同时间存在 ？OK， 同时间存在 meaning like 他们同时间有活性，然后同时间在跑，那样子很奇怪。很好，很好，非常奇怪。为什麼它會很奇怪 ？OK， 它什麼東西都沒做到 ，make sense。但是在 metabolism 上面呢，它還會有另外一個很尷尬的一件事情，是 what happened to 它的 ATP。Glycolysis generates how many ATP? 2, right? Gluconeogenesis consumes how many ATP? 6, right? So both of them, if let's say both happen at the same time, Glucose concentration does not change, pyruvate concentration does not change because they happen at the same time. Let's say, OK， 我们说就是这邊下去的速度跟上面回來的速度一樣快啊，那你 glucose 跟 pyruvate 的量都不增不減 ，OK？ 那 what happens？ OK？Turns out， 如果你同時兩個都 on 的話，你會不斷的在消耗 ATP，right？ 你下去做兩個，你上來吃6個 ，right？ 所以你會 total net， 你會在燒 ATP。Okay，so this is what we call a futile cycle. Okay, why? Because glucose to pyruvate, and I produce 2 ATP. Pyruvate back to glucose. I need to consume 6 ATP. Right? So basically, the net is equal to minus 4 ATP. So I did nothing but consuming 4 ATP. So that's not a good thing. Okay? And there are some enzymes that are controlled very quickly. Obviously, control, remember we talked about control? Control happens at the steps with large delta G. Okay, so all the phosphatases and the kinases are going to be controlled. Okay? And go home and take a look at this slide. They're going to be controlled by something that is going to be making a lot of sense. If you need energy, it's going to run glycolysis. Okay, so for example, if you need energy, it's going to do this. So it's going to sort of inhibit the reverse. Okay, so you need energy, you have less ATP, more AMP, so AMP inhibit this guy. Okay, now if you have too much energy inside the cell, now you're going to inhibit glycolysis because you have too much ATP. So ATP is going to inhibit glycolysis. Make sense? So depending on the energy state inside the cell, does your cell have a lot of energy, not so much energy? You can decide which direction your pathway will go. Okay, now, this is a very specific example of PFK and FBPase activity happening on the same enzyme. Okay, so basically, this enzyme has two states, like the T state and the R state that we talked about back when we talked about hemoglobin, right? Okay, so pretty much the same idea. You have one state that's like the T state and the other state that's the R state, okay? And they're going to respond, okay? The enzyme will respond to the presence of activators and inhibitors, okay? Through allosteric interaction, right? And so very briefly, just go over this again. This is an example of signal transduction from outside the cell, okay, which can control glycolysis and gluconeogenesis on and off, okay? Typically, outside the cell, you have a lot of molecules flying around, okay, including glucose, including hormones, okay? And in this particular example, okay, you have this enzyme. Okay, and this enzyme can be repressed by a couple of different things, and in particular, I want to talk about this F26BP. Okay, F26BP has nothing to do with F16BP. Okay, so it has nothing to do with metabolism. Okay, it's really just a regulator molecule. Okay, it's nothing more than a regulator molecule inside the cell. Okay, and what this F26BP will do, let's first talk about how you make it. You make it from fructose 6-phosphate, F6P, and then you have an enzyme that's PFK2, which is pretty much the same type of enzyme as your phosphofructokinase, but it's specific to activate C2 instead of just the normal F16BP. Once you do this enzymatic reaction, you produce F26BP, which then can in turn regulate the PFK and FBPase1. PFK, let's say PFK1, we call that this guy, okay? This guy, we call this PFK1 and FBPase, okay, like 1, okay? So these two. And what you get is, if you then have F26BP, that is going to start regulating these things, okay? It's going to start regulating these things. Right? And basically, what happens is that your PFK2. Okay, so this guy is the PFA2, which synthesizes this thing. Okay, either it goes this way if it's in the T state or the R state, I forgot. And then if it's when it's turned into this state, then it's going to do that reaction. Okay, it's a bunch of arrows flying around. I know, it's kind of ugly. Let me rephrase this again, okay? So we agreed that your phosphofructose kinase, okay, this enzyme, and FBPase, this enzyme, okay, those two are very important steps in glycolysis and gluconeogenesis for regulation. Yes? Okay, we agree, right? And obviously you can see here, there's a specific molecule that can activate glycolysis. This F26BP can activate glycolysis, and it can inhibit gluconeogenesis. Okay, so this molecule essentially says, I want glycolysis. Okay, so then here is how is this molecule made. This particular molecule, F26BP, okay, is synthesized by this enzyme called PFK2 and FBPase2. Okay, so these two domains are on the same enzyme, and this enzyme is responsible for these two reactions. This enzyme, okay, has two states. It has a T state and an R state. Okay, I don't care which one you call which, but basically you have state 1 and state 2. This is state 1, state 2. Right? Now, state 1 and state 2 changes, okay, it interconverts based on whether or not you have a phosphate on the kinase domain. Basically, what this thing does then is this particular enzyme, okay, this enzyme, whether or not it synthesizes F26BP is then determined based on whether your enzyme is phosphorylated or not. If you want to produce F26BP, okay, then you want the active kinase. Let's see how this will work. This particular specific example is given for you want to do gluconeogenesis, okay, because in the blood, your glucose is low. So if glucose is low in the blood, you want to produce more glucose such that the other tissues can use it. Okay, so this is going to happen in the liver. So then, obviously, your liver wants to then make glucose. Okay, so how does that happen? Basically, what happens is your cell is going to sense a low glucose level, and this cell will start to produce, okay, secrete a molecule. Okay, this is actually a protein molecule called glucagon. This is a hormone. Okay, it's a molecule that sort of controls things. Okay, so it's a hormone that can, fly around in your body. This particular hormone, okay, so let's say this is your cell. So basically what happened, this is cell membrane. So this is the cell membrane. And some cell is going to make this glucagon molecule, and what this molecule can then do is it's going to hook up to a particular, this guy is a receptor, or okay, it's some kind of protein that's on the surface of the molecule that can sort of interact with hormone stuff. Okay? And what this guy is going to do, okay, this guy, essentially it's going to activate a particular enzyme that is going to convert a little bit of ATP to this thing called cyclic AMP. Okay? And cyclic AMP, we know that ATP, let's just call this A. Here you have the ribose ring, and then here's like the basically the first P, second P, third P, right? Okay, and obviously you have these oxygens and whatnot. Okay, this guy is ATP. Cyclic AMP is very simple. You know that AMP is okay. This is AMP. So what do you think cyclic AMP would look like? Anyone? Cyclic AMP. Okay, cyclic AMP is going to look like this. So again, this is A, and I'll have my ribose ring, okay? And pretty much this is going to, sorry, this is always attached to an O, by the way. O, P, okay? And then you have another O here, okay? Remember that sugars also always have these A's attached to here. And then your whatever phosphate, okay? This guy, because now this is cyclic, it's connected within the same molecule. This thing is cyclic AMP, okay? Cyclic AMP is responsible for a lot of intracellular regulation. Cyclic AMP does not just leave the cell and do crazy things. It's always inside the cell. Okay? It sort of serves as a intracellular molecule that can sort of interact with regulator proteins and or proteins. Okay? All right, does that make sense? So cyclic AMP can inhibit enzymes, can activate enzymes, things like that. Okay? Now this is sort of intracellular, so it's inside the cell and your hormones outside the cell. Okay? So from a signal that's outside of the cell that can go inside of the cell, sort of you are translating a message from outside the cell to inside the cell. Right? It's like translation. This is like somebody speaking Chinese, and this is like somebody speaking English. So you have to have a mediator in the middle that can translate. Okay? Because you can't just use the same molecule, go in and out. Right? So once you make this cyclic AMP, now you can start to do a couple of things. Okay? This cyclic AMP is going to increase enzyme phosphorylation. So it's actually going to turn up. Okay? It's going to upregulate, turn on an enzyme that's going to do phosphorylation. What is that enzyme called? Here's a question for you. Okay? Here's a question for you. You're going to do enzyme phosphorylation, meaning I'm going to make this enzyme phosphorylated. Okay? So basically, cyclic AMP activates an enzyme. This enzyme phosphorylates this guy. What is that phosphorylating enzyme called? Kinase. It's going to be some kind of protein kinase. Okay? It's going to be a protein kinase because it's a kinase because it's going to phosphorylate something. And what does it phosphorylate? It phosphorylates a protein. Okay? So it's a protein kinase. Okay, that's good. All right. So once you do the phosphorylation, you're going to basically see how it's going to use ATP and then turn this guy into this phosphorylated version. And this phosphorylated version, okay, what this guy then can do is it activated FBPase. Okay, so this is FBPase. Okay, and it inactivated PFK2, which is this guy. Okay, so because you have a signaling molecule inside your blood that says, hey, I want to do some glucose making, okay? So it's going to translate that into cyclic AMP. Cyclic AMP is going to say, oh, let's do kinase, okay? So kinase then phosphorylate your protein, and in this phosphorylated version of this protein, okay, it's going to have the FBPase activity and not the kinase activity. Okay, so the kinase gone. Okay, so kinase gone. Okay, so kinase gone. Okay, so only your FBPase is left. Okay, now FBPase, okay, is going to then start to hydrolyzing stuff. Okay, it's going to hydrolyze away your, where's my cursor? It's going to hydrolyze away your F26BP. Why? Because now you activated this FBPase2, which is this guy, right? And then it's going to go this direction. Okay? So then you're converting this F26BP back to fructose 6-phosphate. Okay? And, well, what happens? So if F26BP is gone, okay, so I don't have this guy anymore. This guy is gone. So this guy's gone. Okay? What do? Okay? You're going to go gluconeogenesis, right? Because you no longer have the inhibition. Right? So inhibition gone. So you're going to do gluconeogenesis. And if you do gluconeogenesis, okay, basically you activate FBPase. This is talking about FBPase1, this guy, right? And then you have increased gluconeogenesis activity. Okay? Makes sense, right? Right. Anybody have any questions on this sort of how you translate message from a intercellular, okay, so outside the cell, some kind of message from outside the cell into a specific action. Okay, this is like a specific action that we want to do. Okay, and how that action is translated. Right. So this is called signal transduction. Have you guys heard of this before? Signal transduction? General biology, maybe. Maybe, kind of, yes. So signal transduction is very important because when you have multicellular organisms, your different cells have to communicate, okay? And this is how they communicate.